Welcome back to Miss Fountain channel. In this session, you're going to learn about tissue preparation for light microscopy, and this is specifically in histology. We're going to begin with a brief introduction. The human body consists of two basic components, that is cells and extracellular materials, which are the products of cells. Histology is primarily concerned with the microscopic examination of the above, that is the cells and the extracellular material, and how they are organized into various tissues and organs of the body. In the preparation of specimen in histology for observation using a light microscope, we have various steps. In here we have 6 steps. To begin with, we have tissue sampling, we have fixation, we have dehydration and clearing, we have embedding, sectioning, and then the final step is mounting and staining the sections. We're going to begin with tissue sampling. In tissue sampling, we find that tissue blocks, that is the tissue samples that are cut into 1 centimeter each in dimension, that is in each dimension, the width, the length, may be obtained through biopsy, that is where there is diagnostic sampling, through surgical excision, or even through postmortem dissection. In tissue sampling, to avoid misleading structural deterioration or even postmortem samples, they should be taken as to avoid deterioration of these tissues, they should be taken as soon as possible. And to minimize tissue distortion, the dissection instrument should be kept extremely sharp. When you use a sharp instrument for cutting, it's not going to distort the cells compared to when you're using a cutting instrument that is not sharp. The excised organ or sample must be immersed as soon as possible after removal into a reagent. And this reagent is what we call a fixative. And that takes us to our next step, which is fixation. Fixation refers to the treatment of tissue with chemical agents that not only retard alterations of tissue subsequent to death or after removal from the body, but also maintain its normal structure. Fixatives act to preserve the cells and extracellular substance of tissues or even organs, and prevent autolytic, that is the degenerative changes, from taking place. Some of the most common fixative agents in light microscopy that are used in surgical and the general pathology or even biomedical research, we have 10% neutral buffered formalin, 4% aqueous solution of formaldehydes. Fixatives cross-link proteins, thus maintaining the lifelike image of the tissue. The resulting coagulation of tissue proteins has a hardening effect on the soft tissues. We find that fixatives also lock into position a number of carbohydrate and fat containing macromolecules that otherwise would be lost during tissue processing. Fixation needs to be rapid enough to curtail release from dead cells of enzymes capable of digesting tissue constituents. Our next step is dehydration and clearing. We start with dehydration. Find that the collected tissues that are, once they are fixed and dehydrated in graded solutions of alcohol or other dehydrating agents, large fraction of the tissue is composed of water. So they're dipped into a graded series of alcohol baths, beginning with 50% alcohol and progressing with graded steps to 100% alcohol. That is to dehydrate, to remove the water. Like you can start with 50% alcohol. The next one is 60% alcohol, 70% alcohol, 80% alcohol, 90% alcohol, until you get to 100% alcohol to dehydrate, that is to remove the water. Then following the removal of the majority of the water from the specimen corrected, that is during dehydration, the tissues are cleared. And clearing is a process of removal of dehydrated agent and replacing it with a fluid that is miscible both with the dehydrating agent and with the type of embedding medium chosen to make the tissue sample firm throughout. Okay, as with the dehydrating agents, we have a number of clearing agents, and the selection of which is dependent largely on the embedding medium chosen. For instance, xylene is routinely used for clearing tissues, and the alcohol permitted block is passed through several changes of this solvent to replace alcohol with xylene. In paraffin embedding, tissues are treated with xylene or even toluene, chemicals that are miscible with the melted paraffin. And this is what we call clearing, since the tissue becomes transparent in xylene and other clearing reagents. It is important to note that in order to distinguish the overlapping cells in a tissue and the extracellular matrix from one another, histologists must embed the tissues in a proper medium and then slice them into thin sections. For light microscopy, the usual embedding medium is paraffin. Paraffin embedding replaces water with paraffin wax, enabling the block to be cut readily. The xylene permitted block is passed through several changes of warm paraffin wax, which is soluble in xylene. In the next step is bending. The tissue is placed in a suitable container of melted paraffin until it is completely infiltrated. Once the tissues become completely saturated, melted wax occupies spaces formerly occupied by water. After the tissue is impregnated with paraffin, it is placed into a small receptacle covered with melted paraffin and allows to harden, forming a paraffin block containing tissue. That is embedding. You find that paraffin is not water soluble. Water is removed from the fixed tissue by passing it through successful stronger solutions of either alcohol, that is now the dehydration part, till you come to embedding. We have machines for this, for where these stages are conducted automatically. Like now we have an automatic tissue processor. Our next step after embedding is sectioning. Here is sectioning. After the formed paraffin blocks together with the contained tissues are trimmed of excess embedding material, they are mounted for sectioning on a cutting device called a microscope. Microscope is just a cutting device. Yeah, when the sectioning is performed by a microtome, which is it's like a machine that is equipped with a blade and an arm. Just when it advances the tissue block in specific equal increments. For light microscopy, the thickness of each section is about 4 to 7. The thinner the section, the less the likelihood that the components will appear superimposed, and that the optimal thickness is about 5 to 8, which is less than the diameter of a typical cell. That's where microtome is used, so that to cut these very minute pieces. And after sectioning, okay, before we continue, I think that sectioning can be performed on specimen that have been frozen in either liquid nitrogen or even on the rapid freeze bar of a cryostat. These sections are mounted by the use of quick freezing mounting medium and sectioning at sub-zero temperatures by means of a pre-cooled steel blade. The sections are then placed on a pre-cooled glass slide, permitted to come to room temperature since they were initially frozen, and stained with specific dyes or treated for immunochemical studies. Immunocytochemical studies, that is. The next step is thinning. And then many tissue constituents have approximately the same optical densities, and therefore they must be stained for light microscopy. Aqueous solutions are usually used in staining, and prior to this, the wax must be removed or dissolved and replaced with water. And this is usually accomplished by passing the slides together with their mounted sections through xylene or toluene, remove paraffin, and then through descending streams of alcohol to remove alcohol solutions to water, as most of the dyes used are in aqueous solutions. Although various types of stains have been developed for visualization of many components of cells and tissues, they may be grouped into 3 classes. Stains that differentiate between the acidic and basic components of the cells. You have specialized stains that differentiate fibers and components of the extracellular matrix. You have metallic salts that precipitate on tissues, forming metal deposits on them. The most commonly used stains in histology are eosin, that is hematoxylin, and eosin, H&E. Hematoxylin is a base that preferentially colors the acidic components of the cell bluish tint, because the most acidic components are deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. The nucleus and the regions of the cytoplasm rich in ribosomes stain dark blue. These components are referred to as basophilic. Eosin, on the other hand, is an acid that dyes the basic components of the cell in pinkish color. Because many cytoplasmic constituents have a basic pH, regions of the cytoplasm stain pink, and these elements are said to be acidophilic. Many other stains are also used in the preparation of specimen for histological studies. And some of these include hematoxylin and eosin, those ones we've already talked about. We have Mason's trichrome, which forms dark blue in the nuclei, red mesokeratin cytoplasm, light blue mucinogen and collagen. We have osin that forms brown and elastic fibers. We have Wigat's elastic stain that forms blue and elastic fibers. We have silver stain that where you'll see a black color and where there is reticular fibers. We have iron hematoxylin, black color where there are striations of muscles, nuclei, erythrocytes. We have periodic acid chafe. You'll see magenta color with its glycogen and carbohydrate rich molecules. We have bright and gem stains. This is used for differential staining of blood cells. Pink, it will turn pink. We have erythrocytes, eosinophil granules. It will be purple. We have leukocyte nuclei, basophil granules. It will be blue. You have cytoplasm of monocytes and lymphocytes. After staining, the section is passed through alcohol solutions of increasing strength, absolute alcohol and xylene. It's then covered with a mounting medium dissolved in xylene. That takes us to mounting. The cover slip may be permanently affixed by the use of a suitable mounting medium. The cover slip not only protects the tissue from damage, but also is necessary for viewing the sections with a microscope. And find that this medium minimizes reflection of light passing through the section. Histological sections are thin, flat slices of fixed and stained tissues or organs mounted on glass slides. That is then followed by viewing. That is basically it, and that is the preparation of tissues for viewing using a light microscope in histology. That's basically the steps that we have. Thank you for watching. Stay tuned for more.